welcome to this presentation about chemistry of drugs and medicine. So we're going to discuss the difference between drug and a medicine, placebo, therapeutic window, tolerance and side effects, the structure of drug molecules and the difference between them, the functional group modifications and how they affect drugs, organic compounds and inorganic compounds. We'll touch more on that when we get to those slides, drug detection techniques and also how computers are used in drug design. So first things first, what's the difference between a drug and a medicine? So a drug is a chemical substance that produces a change in biological function, so it can be pharmaceutical, psychoactive or recreational in nature. So example would be coffee, nicotine or even cocaine. So they can be used to treat, cure and prevent disease. However, they're not all safe and approved. They can have side effects which can be quite mild to severe. So there can be psychoactive reactions, so mood changes, behaviour changes, impaired judgment, addiction, psychological changes, so hormone fluctuations, increased heart rates, decreased blood pressure. They can also obviously give positive side effects, so feeling of relaxation, alleviating symptoms. Whereas medicines, on the other hand, are drugs that have been specifically designed and tested and developed for therapeutic purposes, for treating a patient and producing beneficial outcomes. So for instance, paracetamol, antibiotics, insulin. They've been regulated and approved, so they go through a lot of testing and clinical trials before they're released to the public. They can be reactive with other foods or other medicines. So for instance, if you just generally take a steroid, it can lead to water retention. That's a common side effect, but the main thing to know is that all medicines are drugs, but not all drugs are medicines. So placebo is an inactive substance or other intervention that looks the same, is given the same way as an active drug or treatment that's being tested. So for example, a patient may feel like it's given them a benefit even though it's not got a therapeutic purpose, it's not designed for that. Researchers think it's something to do with chemicals being released from the brain, so endorphins and dopamine, which produces a feeling of improvement or a feel-good effect. There's a trial which I've linked at the end for you as well to take a look at about sorafenib, which is a placebo as well, which patients took after renal cancer. It's a very interesting article if you want to give that a read. The therapeutic window is the peak dose range for a patient essentially. So it's a dose range of a drug that provides safe and effective therapy with minimal effects. So a low therapeutic window dose would mean that the dose is too low, it's not given the effect it needs. Within the window means it's at its peak effect with acceptable side effects, it's doing its job. And above the therapeutic window means that the drug concentration in the blood's too high, which can lead to obviously toxic or harmful side effects. Penicillin has a wide therapeutic window, whereas warfarin has a low therapeutic window, which means it takes more management from a doctor to patient. Tolerance is a decrease in response to medication or a substance where it's been taken repeatedly over a period of time. The body as such recognises it, so it doesn't produce the effects that you want. And then also side effects, which we've touched on before, is an effect of a drug or any other type of treatment that is additional beyond its desired effect. So a lot of the time when you take drugs, they may reduce in hormonal fluctuations, headaches, stomach pain, nausea, acne, that sort of thing. So in comparing drug structures, we need to look at, I've put two very common drugs here, which I'm sure we've all taken from time to time. We've got ibuprofen and paracetamol. Ibuprofen's on the left and paracetamol's on the right. So they both start very similarly with the benzene ring, which is here and here, which we can see. The difference with ibuprofen, it's got a propionic acid chain, which makes the molecule a lot larger. And it also has an isobutyl alkyl group, which also makes the molecule larger too. The difference between the functional groups is that ibuprofen is a carbolic acid. It also has the alkyl group, as I've mentioned before. The main purpose of the drug is obviously anti-inflammatory purposes, because the carbolic acid, it binds to the COX enzymes, which produces obviously that anti-inflammatory purpose of the drug. It's also more fat soluble too, so it stays in the tissues for a lot longer, and it'll stay in the body for about 6 to 8 hours. Whereas in the hand with paracetamol, it's a hydroxyl group and an acetamide. So generally this drug, it targets the central nervous system, blocks pain receptors, that sort of thing. It targets pain signals more. It is soluble because it's hydroxyl, so it binds to hydrogens. which means that it's more soluble, but it does metabolize a lot quicker, so that it stays in the system a lot less time, so more like 4 hours-ish. So the purpose is obviously very different between these two drugs, and it's dictated by the chemical behaviour, which is the functional groups. Their substituents are very similar, but it's important to still look at them as they can still differ from the functional groups. The 3D shape of the molecule, which we'll go into more detail on later, and also the general size and shape of the molecule generally. So the functional group modifications, as we've just spoken about very briefly, dictates the chemical behaviour of a drug. So it's a group of certain atoms within the molecule that have their own characteristics. So a hydroxyl functional group, for example, will make a molecule more soluble, and because it will, as I say, form hydrogen bonds, so it's more absorbed and excreted a lot easier. Functional groups affect pharmacodynamics, pharmacokinetics, and also the safety and side effects of the drugs, which essentially just means how the drugs work in the body. So do they target pain receptors, enzymes? Are they dissolved in fat or water? How quickly they metabolize? And obviously safety and side effects, what effects do they have as a consequence of taking that drug? So I've given you an example here of salicylic acid and what we now know as aspirin. So the main problem with salicylic acid that it irritated the stomach lining and caused a lot of gastrointestinal upset, so produced ulcers, irritation, that sort of thing. It's because of the free hydroxyl group in this molecule. So to counteract that, they modified it so that they added an acetyl group to the hydroxyl group, which was free basically, and it provided like a barrier or preventative line to stop the salicylic acid, which is obviously very acidic, from irritating the stomach lining. So by modifying this drug, now it continues to still block the COX enzymes, bind to them, that cause pain and fever and inflammation. So it doesn't alter the effectiveness of the drug. They've modified it, but it's still just as effective as it was before. But it's obviously a lot less acidic now. It can be taken by patients over long periods of time with minimal risk to health, so causing gastrointestinal problems. So now we're going to look at cis and trans isomers, and which are basically geometrical isomers. So they're basically compounds with the same molecular formula and connectivity of atoms, but they differ in spatial arrangements of substituents around a ring, double bond, or metal complex. Most of the time with organic compounds, it's round a double bond, sorry. So as we can see here, we've got two double bonds, carbons. An inorganic compound is a platinum metal center, which in this case is potassium. So these things can occur in both organic and inorganic drugs, so in these forms. So cis essentially means identical substituent groups on the same side of the bond or the ring. Trans is identical substituent groups, so they've still got to be identical on the opposite side of the bonds or rings. So I've given you two examples for organic compound and an inorganic compound. So they can behave a lot differently, even though the atoms are the same. Some isomers are more effective, whereas some are not. So for example, we'll go through the one on the left here. So for instance, cis isomers as well, they tend to be more polar, which means if they're more polar, they have a higher boiling point but lower melting point because they have more of a tightly packed structure, whereas the trans isomers tend to be less polar, have a lower boiling point and have higher melting points because it's easy to break them up. So we'll have a look here. We can see on the left here we've got the cis butene, which is on the same side of the double bond, which you can see there, and also the trans version of it, which you can see that the CH3 groups are on the opposite sides. So it's important to be able to identify these. So basically, in order to figure that out, we just need to look at the relative positions of the substituents around the double bonds, which you can see here. So here and here, and here and here. So they're on the same side on the one on the left, opposite side on the one on the right. So it's very similar with inorganic compounds as well. So the cis ones, the NH3 ligands, on the same side of the potassium, so here and here, whereas on the trans one, it's on the opposite sides, here and here and here and here, but the molecules are actually mostly inactive. So it's really important to be able to distinguish the difference between these two types of isomers, as one of them can be active and one of them can be not active, like at all. So this brings me on obviously to the significance of the drug function, so pharmacological activity. So one isomer, as I say, can be more active than the other. They can be metabolized a lot differently as well, so and distributed and absorbed and eliminated differently by the body, so one may take well, one may not. So also toxicity, so that's a really important factor. One isomer might be safer where the other might cause a lot of side effects, even just by very so slightly different arrangements of the substituents. Receptor interactions, so the shape affects the binding and receptors of the enzymes too, so it really does affect that as well. Chemical properties, so cis isomers are more polar, as I've said before, whereas trans ones have a higher melting point. So that's some of the main things to look out for. But I've put also a link at the end so you can have a look at some more examples so you can get a better understanding of it. So we'll look at chiral molecules and optical isomerism as well. So we'll have a look through these now. So I've given you 3 examples. So chiral molecules are very similar to 3D objects, that they don't match the mirror image. So if you look at both your hands, one doesn't match the other, sort of thing. So they're both, chiral molecules are carbon atoms attached to 4 different groups, which create a mirror image forms called atomers. So they have atomers have identical or atomic connectivity but different chemical structure. So that's really important to know. So I'll go through these with you now one by one. So with the first example, we've got 2-propranolol, which is a acyryl drug molecule. Sorry. So the reason that it's this is because it's got a central carbon still. It's bonded to 2 identical groups, and we know that the criteria is 4, so we know automatically that it's not a chiral molecule. It doesn't have the atomers as the mirror image of 2-propranolol is identical to the original molecule. So if the molecules are identical, that means they're going to behave the same biologically. So we know that it's not a chiral module, it's an acyryl molecule, because the mirror image is the same, so they're going to perform the same biologically. With the second example, this is 2-butanol, which we know is a chiral molecule. The reason it is because it's bonded to 4 different groups. So it has 4 different groups containing a chiral centre, as we did on the other one, but the difference is that there's 4 which meets the criteria, so it's important to look at that mostly. So it has 2 atomers which could potentially bind differently to different enzymes or receptors. So it can produce different biological effects. So a chiral molecule can produce different biological effects, not the same. So that's why we know it's chiral. So I've also given you a last example, which is ibuprofen, which we've spoken about recently as well. So it's bonded to 4 different groups again, which we can see. I've linked it at the end so you can have a look at the structure of that molecule in more detail. It's got 4 different groups, it's got a chiral centre. So it has 2 atomers, ibuprofen. So I've put them here, so there's S-ibuprofen and there's R-ibuprofen. So it's really important, and this is why we need to make sure that we understand this, because S atomers are active, and that's the anti-inflammatory effect, whereas the molecule that is very similar to it but not quite the same, it's less active, so it's called the R-ibuprofen, but the molecule is still chiral, and it's because it's got 4 different groups, so that's why that happens. So the general takeaway as well is that it's really important to know how to distinguish these things in the body, as different atomers can act differently. So one might be more effective, one might be more active, one might be more safer to use than the other. So it makes drugs and obviously drug research a lot more safer and effective generally. So now we're going to have a look at techniques for drug detection generally. So there's a few different ways we can look at testing samples. So we can look at biological sample testing and also testing of actual substances. So first I'll go through biological sample testing with you and how these work. So we've got blood tests firstly, and also it's important to just have a quick note that a biological specimen is a material from a living organism which can be analysed in a lab if it needs to be. So the example, urine, blood, saliva, hair, sweat to detect drugs in workplace settings, clinical settings, forensic settings, and the results generally are available a lot quicker than what they are if you send them off for sort of testing substances. Whereas sometimes urine and saliva ones may take a bit longer for others, and they also differ in how specific they are. The choice of test basically just depends how long ago the drug was taken, how invasive you want it to be, and the analytical goal generally. So a urine test, the blood tests are to measure drug concentration at alcohol levels. The results are generally reliable up to 24 hours. There's something called microsampling, which is slightly less invasive than normal blood testing. It's a lot of the time it's used in forensic investigations, such as for driving under the influence or post-mortem inquiries as well, so for toxicology, which you might have heard of. There's also saliva testing. They're very minimally invasive. I put you a little image in there so you can see how it works. Best for detecting drug use within 4 to 10 hours usually is the window, and convenient for on-site testing. So for example, police officers might use it for roadside testing, and it doesn't need laboratory facilities as well, so it's a lot more effective in getting the results a lot quicker. It's a lot more cost effective too. And then also now we've got urine tests. These are probably the most common, the non-invasive method for it basically just uses antibodies to detect drugs in the urine, typically 24 to 48 hours after drug use. They use these sometimes in intermittent drug test screenings in work, so they're very common to use. Hair testing, not as common, but the mummy can detect drug use over a longer period of time, which is typically 90 days depending on the length of the hair actually. They can be used in forensic or legal cases where repeated or long-term drug exposures need to be proven. And then there's also something called sweat testing too. It's a lot less common, as you might imagine, than the other ones. They're used to detect drugs over weeks using these adhesive patches onto the skin. They might be used in sort of rehabilitation cases, that sort of thing. It can be trapped over weeks without having to redo urine, blood, saliva testing. So next we're going to look at chemical analysis of drugs, so actually looking at substance testing. So for example, I've given you two here, which is the most common, which is presumptive blood testing, which is more for quick screening, and confirmatory blood testing, which is more definitive identification. So with presumptive blood testing, it provides more of a rapid indication of presence of a drug, whereas with confirmatory blood testing it provides a more definitive identification, and also the quantity of the drug within the substance too, but it just takes a little bit longer. There's different methods of presumptive blood testing. They can use something called colorimetric testing, where a region is added to a sample, which is like a chemical or substance is added to another substance or a sample, so for example urine, and it'll change colour if it tests positive for a certain drug. Sometimes they might use it at airports to security and things like that. It doesn't quite give you a quantitative sort of result. It doesn't tell you how much of a drug there is, it just tells you whether it's present or not. So that's one thing, and they also do tend to use them more in field testing or like a lab screening. The benefits to it is that it's not as expensive and it's a lot quicker results, but they're not definitive. So as I've said, there's colorimetric testing and urine dipsticks, urinalysis that they may use. There's also different types with confirmatory blood testing, which is a little bit more in depth. So we've got the GCMS, which stands for gas chromatography mass spectrometry. LCMS, which is liquid chromatography mass spectrometry. And then we've also got thin layer chromatography as well, which is this one, and also general spectroscopy. Excuse me, generally as well. So gas chromatography is a type of confirmatory blood testing, as we've touched on. It basically separates compounds by vaporising them, and then they move through a column each. Each compound comes out at a different time, so that the mass spectrometry measures the mass of each compound fragment so it can actually identify exactly what it is. Liquid chromatography, similar, but it's not vaporised is the difference really. So it's done for drugs that may break down when they're heated up. So it also does identify exact compounds in a similar way, but it's for basically testing without vaporising the drug and eliminating it. Thin layer chromatography is basically placing a small sample on a flat plate coated with silica. A solvent then moves them down the plate, carrying compounds at a different rate so that they basically separate. It's then put under a UV light, and the chemicals will change colour so that they can be identified individually on this plate, which is very clever. The spectroscopy basically analyses the structure of the molecules. It looks at the molecules on a molecular level rather than what we spoke about already. And now we're going to talk about drug design. So there's quite a lot of methods, as I've put here, of detecting sort of new drug molecules and identifying them. We will go over a few more in specific detail. So high throughput screening, structure-based drug design, ligand-based drug design, virtual screening. So for example, target identification and validation finds and confirms biological molecules, so like proteins, enzymes, that are suitable targets for new drugs. Public databases are also useful, as they've got obviously biological, pharmacological data to inform drug discovery too. And the ligand-based drug design as well designs new molecules based on structures of active compounds as such, so that's really useful to use. We'll go in a bit more detail for obviously identifying new drug molecules specifically. So I've put a definition of what drug discovery is, so you've got that. So essentially, drug discovery, the high throughput screen is a very useful one. So it rapidly tests millions of compounds using automated equipment against a chosen biological target. So they test it against a specific target, such as an enzyme or a receptor. So an example, so it targets sort of active compounds, so they're called hits. They're then optimised or changed, developed into potential promising drug candidates. So an example is that high throughput screening was used to find molecules that block HIV protease. I've also linked that at the end for you, but basically HIV protease is an enzyme, so if HIV becomes infectious, by blocking the enzyme, it stops the replication, and HSS are allowed, which is the benefits for rapid identification of suitable drug candidates for curing this or combating this effectively, and basically block it. And then we've also got structure-based drug design, which uses a 3D structure of a biological target, again like an enzyme or receptor, design molecules that will fit precisely, sort of like a lock and a key sort of scenario. The molecules are then tested and optimised again as potential drug candidates. So the benefits of it, it shows the chemical features of the target's binding sites. So for example, with this, they use imatinib chronic, so CML is what it's basically short for. This was created using this technique, it's obviously a lot more, the structure-based design's a lot more highly selective, and effective, which obviously is a lot more useful to research when trying to find the correct drug to use as a candidate, and it helps a lot of get rid of a lot of wasted efforts by scientists and researchers. There's also virtual screening, which uses computer algorithms essentially just to search through large volumes of compounds and predict which ones are most likely to bind to the biological target. So very similar to the other two, but it searches through a lot of large volumes of compounds. It saves time and money by narrowing down candidates before laboratory testing. So virtual screening was used to identify compounds that bound to the SARS protease as well. So virtual screening predicted compounds that could bind basically and inhibit them. And then we want to have a look at computers generally in drug design. So I put the definition up there that assists scientists in identifying and optimising potential therapeutic compounds by using computer power and biological knowledge to reduce the time and cost of drug development, which is a very big one. So computers basically make drug design a lot faster and more accurate. So they can simulate molecular docking, narrow down compounds that are actually worth testing rather than wasting the time, and predicting whether or not they'll be safe and effective before a lot of expensive lab work is carried out. So I've given you a list here. So molecular modelling uses 3D models of molecules at targets, visualises interactions at molecular level. Molecular docking predicts how well a drug will fit a protein. So with that, and then there's virtual screening, as we've already touched on, so it uses algorithms to search large chemical databases to find compounds that are likely to bind a target. So it narrows down the drug candidates, saves time, resources, and focuses lab experiments on the most promising drug leads. Quantum computing basically uses quantum simulations to model molecular interactions at an atomic electronic level. So basically it uses quantum mechanics to simulate molecular interactions. It allows scientists to obviously predict potential toxic effects as well, and helps researchers identify safety concerns well ahead of time. And it also obviously reduces the risk of failure if they can do this in advance. There's also pharmacokinetics and toxicity prediction, so ADME, which is absorption, distribution, metabolism, excretion, and potential toxicity of a drug, which enables scientists to identify safety issues and side effects early. So obviously before there becomes any problems in people, they can try and basically analyse how a drug moves through the body by using computer systems, which is really effective for making sure that they're safe for general use, and also saves a lot of time generally. There's also something called QSAR, which uses statistical analysis of compounds that are already known, predict activity of new molecules, analysing the link between chemical structure and biological activity. So basically it just allows researchers to predict the activity of new molecules to guide the design of more effective drug candidates generally. So I put the references on here, just what I spoke about through the presentation for you to take a look at in more detail. And that concludes the presentation. If you've got any questions at all, please let me know and I'll be happy to help. Thanks for listening.